So thank you everyone for joining us today. My name is Eric Tweet. I'm a data wrangler on the Foreign Contamination Screen team at NCBI. And today I will be sharing new tools in development by the FCS team to remove contaminants from genome assemblies, which will improve the genome assembly submission process at NCBI. And there are other FCS team members here today. We have Terrence Murphy, the product owner of the NCBI RefSeq offering, and Deacon Sweeney, the FCS technical lead, who will help to answer questions during the webinar. So an overview for the webinar content today. First, I will go over a brief background on genome assembly contamination and the current pipelines in use at NCBI to detect contamination. Next, I will provide details on the new NCBI FCS implementation that includes publicly available screening tools. And finally, I will run through a live demo to show how a typical user could run FCS tools in the cloud. We define contamination as sequences present in a data set that do not originate biologically from the indicated organism. Contamination can occur at many steps in the genome assembly process, starting from the initial DNA extraction all the way to data submission. And there are multiple potential sources of contamination in a genome sequencing project. In the example of an insect genome, there could be contamination from its host plant and bacterial contamination from endosymbionts or introduced during lab sample preparation. In the context of genome assemblies, foreign DNA can be assembled as complete or chimeric contigs, and ideally, a contamination screening pipeline should be able to identify contaminants from multiple sources and distinguish those from contigs belonging to a user-defined source organism. Contamination detection is important because contaminant sequences have negative consequences when introduced into the NCBI archives. Contamination pollutes the public archives and reduces the statistical power of big data analyses. And there are multiple published reports of contaminated entries in NCBI databases. In addition, scientific conclusions can be inaccurate when sequences are incorrectly attributed to organisms. This paper is a notable example that showed bacterial sequences that were initially thought to be the result of horizontal gene transfer into a tardigrade were most likely to be bacterial contaminants associated with the sample. And the use of secondary data analysis to identify evolutionary conservation can also be affected. As this paper points out, proteins conserved by diverse bacteria are in reality shared human contamination. NCBI has maintained a legacy FCS pipeline for 20 years and is used to screen hundreds of thousands of prokaryote genomes and thousands of eukaryotic genomes each year. Here's a diagram of the steps commonly used in genome assembly, including an NCBI contamination screening step prior to final assembly submission. One pitfall with the current approach is that contamination identified at the time of submission to NCBI can delay the release of the data, and depending on the extent of the contamination, a user might have to go back and redo some of the prior assembly steps, such as assembly, correction, or annotation. Public access to FCS tools allows genome submitters to screen assemblies much earlier in the process. For example, immediately following the contig assembly stage. This allows users to identify and address contamination issues earlier and save the headache of redoing work. And in the future, we will have a system set up to allow genome submitters to submit their clean sequences along with contamination reports to streamline the submission process. Now I want to give a broad overview of the two tools that are currently part of the FCS suite. FCS Adapter is a re-implementation of NCBI's VecScreen pipeline and looks for adapters commonly used in sequencing library preparations. The general workflow involves a FASTA format validation step, a BLAST search step using high stringency parameters to look for exact or near exact matches, a filtering step to remove weaker hits, and a processing step to handle overlapping hits in edge cases. The pipeline produces a cleaned FASTA file that has sequences either trimmed or removed depending on the extent of the adapter matches. The second tool we call FCS GX, which is a brand new tool that looks for contamination from foreign organisms. The workflow here is to split the FASTA and mask low complexity regions, followed by an alignment step that operates in 2 passes. The alignment step uses a large database of diverse genomic sequences from eukaryotes, prokaryotes, and viruses, and higher scoring alignments from the first pass are retained for a second pass of the alignment, shown here in the diagram. Next, alignments are refined and coverage and score information is collected, and the pipeline produces a final report that lists sequences with various contaminant cleaning actions. Users are guided to remove sequences entirely, trim off ends or internal segments, and some lower confidence alignments may be in scope for further review. In the future, we will implement a clean FASTA step similar to FCS Adapter that automatically generates a final FASTA file after interpreting the action report calls. The major goals for the FCS tool suite are that we want the tools to be easy for users to run, we want them to be fast, we want FCS to be reliable, so reproducible across different types of compute environments. We want the tools to be cloud ready, and we've been able to test FCS in both Amazon Web Services and the Google Cloud Platform. We want contamination results to have high sensitivity and specificity. The current FCS pipelines at NCBI miss many contaminants, so we want a more sensitive tool. And at the same time, it's important that we do not remove valid sequences from assemblies, so we want to minimize false positives. We also want to use the same code that NCBI uses for screening incoming submissions to make that process consistent. And we've released components of FCS and have received user feedback on our releases. We've also released new software releases that have addressed some user requests. And here's the HTML link to the FCS GitHub page, which we will visit as part of the live demo. As far as system requirements, you need a Unix operating system and Python version 3.7 and above. If you have root access to your system, you can use the FCS Docker image. Those without root access can use Singularity. And I've listed recommended and requirements in terms of CPU cores and memory for running FCS Adapter and GX. For GX specifically, the 500 gigabyte memory requirement is for holding the large reference database and accessory files. And execution can utilize up to 48 CPU cores. If these requirements are prohibitive locally, both Amazon and Google Cloud Services provide hosts sufficient for execution and can be run at the cost of only a few dollars per genome. So now we're going to transition over to the live demo. I'm going to demonstrate examples of both FCS Adapter and FCS GX. For FCS Adapter, we're going to be looking at contaminants in tsetsefly. I'll show you how you would copy a file from your local computer to a virtual machine. And we're going to run FCS Adapter using the Singularity image. For FCS GX, we're going to be looking at contaminants in AP complex and parasite plasmodium and run using the Docker image. All right. So just to provide you with some orientation here, on the screen up top, I have the set of commands that I will run to get the FCS tools up and running. Some of these I've already done prior to the webinar, and others I'm just going to copy and paste into the terminal to save time. The FCS GitHub has wiki pages that provide a step-by-step process to run the tools. And on the bottom here is the interactive terminal window that I'll be using to execute the commands. I also want to mention that this demo assumes that you have a cloud instance set up. So here I'm already logged into my Google Cloud Platform virtual machine instance. Again, we've tested on both GCP and Amazon Web Services, and some users may be able to run the FCS tools on their local compute farms as well. So for FCS Adapter, we're going to be looking at data from a tsetsefly, and here's the NCBI Genome Assembly page for this particular organism, just showing for reference purposes. The first step would be to download this assembly into my working directory and copy it into the cloud virtual machine instance. So these are the commands right here. So I've already done that. So this curl command retrieves the genome, and this gcloud compute SCP command copies it from my local machine to my virtual machine instance. And Amazon Web Services has a similar command to the gcloud compute SCP. So again, I've already done this. I'm going to go to my FCS Adapter directory and show that I've kind of downloaded this already. So then you would log into your virtual machine instance, and then you would download the runner and the singularity image. So again, I've already done those things with curl commands, making the runner user executable with this chmod command. So I'll just move on to the next step, which would be to run the program itself. I should mention too, for those of you not familiar with Docker and Singularity, the images are essentially a file system and parameters used to execute code with all of the software dependencies baked in. The benefit here is that with each new release of the software, you don't have to worry about compatibility issues, so it's all self-contained in that sense. So to run FCS Adapter with Singularity, you need your FASTA input file, shown here. I've specified an output directory. You use a uke or proc parameter depending on your source genome. So since we're working with that etsy I have specified uke. We're working with the Singularity container engine, and I'm specifying the FCS Adapter Singularity image file .sif that we downloaded with a curl command in the previous step. Now it takes a couple minutes to run this whole genome through FCS Adapter. And so just for simplicity's sake, we're going to run a single sequence, which will be a little bit faster. And so the program is running now. I do want to point out that there's a warning message and a fix the problem message here. This is currently expected output, and so users running this program shouldn't be concerned that something is running incorrectly when they see these warning messages. Again, just as a reminder of an overview of the pipeline, we're first validating the FASTA file, doing a stringent blast search, filtering alignments, and then making a set of final calls based on the parsed alignments. So this log that's printing the screen right now provides a report of the pipeline going through the various steps. And a successful run should print a bunch of completed success messages at the bottom of the screen. So now that we've completed the run, we can actually look in the output folder. So here are the output folder contents, and many of these files are logs or records of parameters used. The important output here is this FCS adapter report.txt. And so we're actually going to look at the results here for the full data set first, not just for the one particular sequence that we ran on FCS Adapter. So you can see that this is a tab delimited file format with the sequence accessions in the first column, the length of the sequence is next, followed by an action. So these are either going to be action underscore trim or action underscore exclude, depending on whether it's suggested that a adapter gets trimmed off the end or in the middle, or if the sequence should be removed entirely. This range provides the base pair range of the match the adapter. And then the final column has more detailed information about the adapter matches. So as I kind of scroll through this file, you can see that the primary matches are to Illumina paired end PCR primers and Rubicon Genomics DNA-seq dual index sequences. Now we're going to look at the specific sequence that we ran, just the individual sequence that we ran on FCS Adapter. So from the overview that I gave from the full file, we can see that this sequence is about 2 KB, and there's a suggested trim at the beginning of the sequence in the first 58 base pairs, and it's matching this Illumina PCR primer. So what I want to do now is go to my internet browser just for comparison purposes. You might not know that NCBI has this web vec screen tool, where you can enter your sequences and search for vector or adapter contamination. So I've already gone ahead and pasted this sequence into this box, and we're actually going to just run VecScreen right now on this sequence. And so you can see the web VecScreen results produces matches that align with what we saw with FCS Adapter. There's a strong match from 6 to 58, and then the suspect origin from the 1st to 5th base pair. And you can see the alignment to this Illumina PCR primer in the pairwise alignment viewer down below. So what this is saying is that there was a strong match at the 6 to 58 base pair interval, and given that it's close to the end of this sequence, it's suspicious. Errors could occur due to sequencing errors, but it's recommending that that entire end of the sequence be trimmed off. And this is what we commonly observe when we see adapter contamination in genome assemblies, is that adapters are commonly found at the ends of contigs. Now, the NCBI web page for this particular assembly doesn't have any details about this particular assembly method, but the ENA equivalent for this tsetse fly does detail that the whole genome shotgun assembly is based on a combination of Sanger 454 and Illumina reads. And so it makes sense why we could observe Illumina sequencing primers in that data. I just wanted to kind of show that. So now I think before we transition over to FCS GX, I should mention that kind of the primary benefit that we see for FCS Adapter is that instead of looking through individual sequences like this on web-based VecScreen, we can screen through entire genome assemblies in a very high throughput manner. So now we'll transition over to FCS GX and look at contamination from foreign organisms. The genome assembly of interest for this demo is going to be an apicomplexan parasite Plasmodium. So I'll go back to my terminal window and go to my FCS GX directory. Okay, so the steps to get this running largely follow the steps for FCS Adapter. We're going to start by downloading the runner and the genome data, which I've already done. The next step is to create a shared memory space. There are various methods of doing this, which may depend whether or not you have sudo permissions on your compute environment, and these are described in the wiki. And these have to do with what I mentioned earlier on the PowerPoint in terms of the shared memory requirements used to store the database files. Optionally, you can also provide a path for a local backup copy of the FCS GX database. So when you run FCS GX for the first time, it will actually download the database from the FTP site, which in our tests currently takes about an hour in North America. And then the actual pipeline of GX only takes on the order of minutes, a minute or two. Once you have the GX database saved as a local backup, the actual process for screening subsequent genome assemblies should be quite quick. So to run FCS GX, we'll run this command right here. I've already downloaded the local backup copy, so we don't have to wait an hour for that to happen. But the requirements for running this pipeline are your input FASTA file. I've specified an output directory. I've specified the path to my shared memory location, and also the location for the saved database files on disk. We also specify the split FASTA parameter to split scaffolds into contigs, and specify this tax ID parameter, which is an NCBI taxonomic identifier number that corresponds to your declared source organism. So this is specific to the Plasmodium species that we are testing, and will help GX differentiate between source organism sequences and contaminant sequences. So I'm going to run this right now. You can see there are messages relating to the the database. I'll just kind of scroll up here so you can see these. It's recognizing that the database files are already stored on disk, so it's going to go right to the screening pipeline. Now at this point, we are doing the masking step. And you can see above are some of the parameters used in the run. Again, just an overview of this particular pipeline. After the masking step is a 2-pass alignment step, followed by some post-processing of the alignments and coverage values to produce a final set of contaminant calls. So upon the completion of a successful run, you should see these contamination summary reports at the bottom of the screen. And I'm going to kind of make the screen a little bit larger so you can see. So the contamination summary report that prints out at the end of the run kind of details the contaminant taxonomic divisions in terms of the sequence counts matching different taxonomic divisions and the base counts corresponding to those divisions. So from the printout here, we see that the primary contaminants are G. proteobacteria and rodent, comprising over a megabase, almost 2 megabases of contaminant sequence. And then there's also an action summary report below that contamination report that gives a list of the total, a summary of the actions that a user could take to rid the genome assembly of contamination. So in this example, almost all the sequences are recommended for an exclude action, which means they should be removed entirely from the genome. And one sequence is marked with a trim action, meaning it was identified that the sequence likely contains some plasmodium sequence, but also contains some contaminant sequence. Now, if we look in the output folder, you can see 2 files that are generated using an FCS GX run. One is the GX report file, which provides the final contaminant cleaning actions. And the second is a taxonomy report file, which is a more intermediate output that users can look at for additional details about the contaminant calls. So we'll look at both of these. First, we'll look at the GX report. So similar to the FCS adapter output, this is a tab delimited file that provides summary information about sequence successions in the first column, the coordinates corresponding to the contaminant ranges in the second and third columns respectively, and then the action is kind of in the middle there. So similar to the action report that we just saw, the action summary report, almost all these sequences are recommended for exclusion. And I'll kind of scroll down and you can see that the predominant matches here are rodent corresponding to mouse, and this G. proteobacteria methylococcus capsulatus. All right. Now you can use some parsing commands to look at more specific sequences of interest. So maybe I'm interested in that one trim call that GX suggested. So I can do a grep command to find that sequence. You could also use grep to find sequences corresponding to certain taxonomic groups, for example. So I have the sequence accession for this trim call, and now what I want to do is look at the intermediate file, the taxonomy report for this particular sequence. The way that this taxonomy report file is organized is it has some summary information in the first few columns about the sequence size, any repeats identified, and the length of regions matching any of the top 4 hits. And then what these pipe symbols separate is up to 4 distinct matches for any given sequence. So the taxonomy report file will report up to 4 distinct matches per individual sequences. And for each taxonomic division, so for example, protist alveolate, it will report a maximum of 2 matches. The advantage here is that if closely related organisms share common contaminants, GX reporting multiple divisions can potentially distinguish between valid contaminants and artifacts due to shared contamination. The double tilde symbols in the sequence identifiers indicate that this particular sequence is assigned as chimeric by GX. So if you look at the results for this first 2,700 bases, you can see that there are two strong matches to alveolates, shown here and here. And the assignment of this particular subsequence is protist alveolate with a coverage of 100%. Then in the next 600 bases, there is one strong match to mouse. And again, it covers that entire span of 615 base pairs, and it's assigned contaminant rodent with a coverage of 100. Now, this is the high coverage information is strong evidence that it is a valid trim, but even more, what increases our confidence about this call is that we know that there are other mouse contaminants elsewhere in the assembly. There were a lot of exclude calls that were rodent, and so that increases our confidence that trim calls are valid as well. Now I'm going to go back to the genome assembly page for this organism and point out that this assembly has an associated publication that indicates that this Plasmodium parasite is a malaria parasite of rodent. So finding rodent sequences in this assembly is unsurprising. And I also ran a blast search of this particular sequence. And you can see that the graphical results correspond to what we observed in FCS GX. So the first 2,700 bases correspond to different Plasmodium sequences. And on the right here, the region that FCSX called as a trim correspond to mouse. So we're highly confident that this is a valid trim. So that's it for the demo portion. The last thing that I want to just give a brief overview of is the FCS GitHub page. This is just navigated to the homepage right now, which gives kind of a broad overview of FCS and some more details about the individual tools, FCS Adapter and FCS GX. I mentioned the wiki pages. I'll just click on the FCS GX wiki page. And so this page gives us kind of a summary information about FCS GX, its prerequisites, and then I'll kind of scroll down here. This gives a full kind of quick start. So how you would download the runner script, how you would get test data that we've tested with just to make sure that the pipeline is working as intended. And then it gives pipeline examples for both Docker and Singularity. And on the bottom, it gives different usage examples, ways to interpret the output, and on the bottom, some known issues. We also have some technical information about the algorithms underlying the pipeline. So please check out the GitHub page if you're interested in more information about that. In our wiki, we also have information about FCS Adapter, the more intermediate FCS GX taxonomy report file. As I mentioned, we have set up documentation for both AWS and GCP. So if you're excited about using this in the cloud but don't know how to get started, this would be a really good resource to use. And finally, we have some release notes that kind of describe any major updates that we have to the software. So as you can see, we've already had a few new releases since the initial release in March, and we've responded to some user feature requests with this method. So please, again, check out our GitHub page if you're more interested about these tools. If you're testing these out and have issues or suggestions, please reach out to us on the FCS GitHub issues page. You can also contact us with questions and suggestions about FCS through this email, refseq-support@nlm.nih.gov. And major updates to FCS are oftentimes posted through the NCBI social media channels, which are shown here. We have a lot of people to thank for work on this project. And the last thing I would like to mention is that FCS is part of the NIH Comparative Genomics Resource, an NLM project to establish an ecosystem to facilitate reliable comparative genomics analyses for all eukaryotic organisms. And I'm happy to take any questions.
Thanks very much, Eric. So we have some questions. One of them is this one, which is, instead of removing the contaminants from assemblies, isn't it easier to remove the contaminants from the reads? And I suppose part of the answer to this is that we're talking about things that are already assembled that we're going to be submitted. Do people find, even after they remove stuff from the reads, that they're still in the assembly?
So this is a good question. And for the adapter specifically, I know that there are tools out there that specifically operate and are intended to be used on reads to trim out adapter sequences. So yeah, like you mentioned, one major benefit for this implementation would be that if a user had already gone through the step of doing an assembly and didn't do like an adapter removal step, then this would be beneficial. And then as far as the organisms, there's just not as many tools out there that specifically operate on reads. And so this is, again, something that we think is new territory in a lot of respects. And it also operates very quickly. And so it has kind of that high throughput component that we're excited about. I also mentioned that we're wanting to make this publicly available to make it, consistent with the process that NCBI is currently using to screen incoming submissions for contamination.
Right, so we're using it mainly on assemblies, and so that's why we have it that way. Okay, so here's some other ones. Can you explain, like you mentioned, two different container, Docker and Singularity. What are the differences or advantages of using one versus the other?
Right, so I think Deacon would probably be the best person to answer this question to provide additional details. But what I can say for starters is that Docker usually requires you to have like elevated privileges on your compute system. So if you're trying to run something locally, if you don't have pseudo permissions on your compute environment, you usually can't run Docker. And so Singularity provides the advantage there, but I think Deacon should probably jump on and clarify any additional points of comparison.
Not at all, Eric. I think that really summarizes it.
Okay, thank you. So another question, and this one is relevant for prokaryotic assemblies. So the phage components, where they're in prophage in the bacterial genomes, how does the contamination screen handle that, or is it just for eukaryotes?
Oh, that's an excellent question. And it is something that we will encounter with eukaryotes too, in terms of lateral gene transfers. So we're currently working on developing a way to sort of handle these cases. Usually we won't report prokaryote viruses as contaminants in prokaryote genomes, and same with eukaryote viruses found in eukaryote genomes. However, we would report, say, a prokaryote virus in a eukaryote genome. But the same is true with like a lateral gene transfer. I mean, we know that there are known cases of this. And so we're kind of working on, do we want to handle these on a case-by-case basis? Is there a way to sort of treat these all similarly? And so this is a content piece that we're currently working on, but it's certainly from a biological perspective, very interesting.
Cool, thank you. So in terms of, I certainly, myself, this is a question from me, basically. So when I run blasters, a lot of times I can identify easily contaminants that are present in Genome sequences. Is there an effort of sort of going back and looking at things before we had this tool, but like going back into the archive and trying to find more contamination?
In terms of what we did previously, or
what's already in GenBank?
Yeah, so one thing that I will point out is, I mean, to test our software and to do kind of validation, we oftentimes do those secondary analyses, including kind of blast searches. And one thing also that's important to note is that whenever you're using a reference-based tool, you run the risk of a contaminated database entry kind of, creating noise in your results. And
Yeah.
so when we were working on developing this tool, one of the things that we did early on was screen the genomes that formed part of the database through the tool itself and, masked, or I guess ignored self alignments. And so in that way, we could identify, maybe database entries that could potentially create confounding results. So we have done a lot of that, both manually and kind of in a semi-automated method to improve the quality of the results.
So that kind of leads into another question that we have, which is the background database for GX. So it must be already cleaned up. It's different than the blast database, for example.
Yeah, so we have made a lot of effort to do clean up, both, like I mentioned, through manual validation and kind of automated pipelines to sort of identify what are going to be very high confidence contaminants that should be removed from the database. I can't claim that it's perfect. It would be really difficult to make that claim. And we're finding, if we find new issues, we plan to release new database versions but the goal here is that we want the database releases to be pretty far apart. And so we want to do a lot of the heavy lifting in terms of the contamination removal up front, and minimize the increments or the timeframes between successive database releases. But of course, as new data gets released, that might improve our ability to identify contaminants, like if they're clean genomes. So we also want to consider including additional genomes that get submitted to be in scope for the database in the future.
Right, exactly. To keep the taxonomic scope up to date.
Yeah, because that's also an area of our own personal interest is, identifying contaminants from maybe those kind of poorly represented taxonomic groups.
Right. You mentioned in terms of thinking of, so there's a couple of things that the GX database, that sounds like that would be really cool for a blast database. It'd be nice if we had it available for that, but that's another topic. Also, you mentioned downloading it from the FTP site. Is it not available on the cloud providers?
We're currently working on that. That's like
Okay.
one of our next steps.
Yeah.
Because we've received some feedback in terms of the download times and are listening to that. And so one of the next steps in the more immediate future is going to be making that available on cloud services.
And thinking about the cloud services, one other question is does your GitHub site have the documentation on, like suggestions for setting it up on, like what kind of instance types you should use on AWS and GCP?
Yes,
like
we on the GitHub page, we provide specific recommendations for GX, as that is the one that tends to be more intensive in terms of the requirements. So if users are wanting to run both tools, if you follow the setup recommendations for GX,
right,
that should be more than sufficient to run both tools.
Gotcha. Okay. And I'll just give you one last question. This is another one that's from me. We sort of cannot figure out why the rodent sequences are in that Plasmodium genome. Where did the Methalococcus sequence come from? Because it's a methanogen, so it's like, a weird thermophilic bacterium. I looked to see what it was.
Yeah, I guess I'm interested in that as well, and I haven't given, I guess, that some more critical thought. So maybe something interesting to look at. But
I mean, does it ever happen that just sometimes is there any kind of contamination that arises from some kind of electronic mix-up when they're trying to assemble data or something like that?
Yeah, possibly.
I don't know, but it's an interesting that organism, sequences are in there because it seems just sort of out there. but that's interesting. I think that's it. So thanks everybody for coming, and the materials for this demonstration and the slides are up on the FTP site. That link is in the beginning of the slides.
Yep. Thank you again, everybody, for attending. And please reach out with questions and suggestions.